That's amazing. I know that
Yeah.
in the late 90s, when, because I obviously do a lot of work with stem cells, obviously, I know that, was it in 1998 where they first isolated human, what was it, human cells, was it, back in 98?
Yeah, and basically, once we immortalize the first like human cell line of the HeLa cells, after we discovered like the PCR and all these reverse transcriptase and all these things, it really like enabled us to develop all these different human cell lines,
Hmm.
and then we could just grow them in the lab indefinitely. Um, it's just that those cell lines are not as reproducible for the actual um, patient cells, because it's a lot harder to grow those in the lab because they're not indefinitely being produced. But now we're trying to go back and see, oh, what are the differences between those initial cell lines that we have generated to like actual patient information. So we have to then validate that again. So it's a big process. So
Of course, can I imagine? Because I
yeah,
did want to ask you actually how it works with different patients, because obviously everyone is different. I mean, what do you
yeah.
do when you say inject stem cells, for instance, into a different patient? Do you have to do several different in-depth tests for them to make sure that when you are injecting with the cells, they actually can take it properly?
Oh, of course. Yeah. So there's all these protocols to be able to make sure that it takes it. And then, when you inject cells, we first tried, in grafting those cells in mice, and it's usually, around 20% or 25% that it takes. And then once, so that's why you have to do a whole experiment, very tedious, with many mice, just so that a few could develop those like tumors, for example.
Mm.
And then we're only going to test on those. So it's, yeah.
Mm. You mentioned tumors there as well. I know that one of the early experiences on rats, I think rats and mice, where they're using undifferentiated cells. Is that correct? Where when you're injecting
Yeah.
those, it would cause, are they teratoma tumors? Is that correct?
Yeah, they're like actually, yeah, those undifferentiated cells are like, or the immature type
Mm.
cells are the stem cells. And so that's what we're trying to do in lab now. I'm trying to really focus on what are those phenotypes, or what are those states, because it turns out that there's not just one state that it could be differentiated.
Mm-hmm.
It could potentially be multiple undifferentiated states, and then they can. It's like a shapeshifter, sort of like they
Mm-hmm.
can become uh, with different stimuli. They can become different things, and so we're trying to uncover what those states are, so that we can like develop therapies to target
Mm.
those. Yeah.
That is what I read up on, that these undifferentiated cells can become literally anything. You put them obviously with work to it. But
Yeah.
when you, am I correct in saying that there's two main cells that you work with? You've got the adult cells and the embryonic stem cells as well. Are they the two main that
I
you
mean,
work with?
well, yeah, like we do those in the lab. Um, I'm not currently doing that, but yeah, the embryonic stem cells versus the like the other breast stem cells. So I'm mainly working with that, like breast cancer stem cells.
Hmm.
And so exactly, it's like they can't quite become anything, but we're really honing in on what they can and can't be. But that's exactly what you touched on, is called like cellular plasticity.
Mm-hmm.
So that plasticity is like allowing it to become, have a tendency to become more things. So yeah,
Mm.
I'm pretty much working with primary cells, which are the patient derived cells, and running the screening on those.
Wow,
Yeah.
that is incredible. It really is. And I want to go back to what
Yeah.
we kind of said earlier, where you mentioned sort of proteins in cells and so forth. From what I understand, I know that every cell has its own protein. And when you kind of isolate sort of the cells, I know that stem cells are obviously they're way more rare than, say, regular cells. So, like, say, regular cells got one every 100,000, is that correct? And
Yeah,
then
it's
bone
basically
marrow
like the
cells?
stem. Yeah, exactly. Like the
Yeah.
stem cells are like maybe account for 1% of the
Wow.
whole population, but you have stem cell population in every organ and every
Mm,
tissue.
of course.
So, and they could be in cancer, they could be recruited to like to a different location a different organ, like in breast cancer, it can stem cells from the bone marrow can be recruited to the breast, or the brain, or the lung, and that's what enables the cancer metastasis. When it gets really bad, then it can those stem cells can be also recruited different parts of the body, and then it's like spreads. So we're trying to understand that as well.
That's definitely,
that part is so complicated.
Of course, from what I understand as well, is that you can kind of isolate or find these specific cells by some kind of fluorescent light or so forth. Is that correct?
Yeah, we did confocal microscopy.
Okay.
There's a whole heap of them, but fluorescence microscopy. So we do that by we can isolate it by staining, a staining procedure, but you have to then fix the cells. You have to isolate them first and section them, and then once you section them, then you stain it for a particular protein or whatever antibody, and then you can look at that particular one under the microscope, and it's really high powered. It's extremely amazing the type of level of detail that you can find with these imaging results.
Of course, and I can know that obviously with stem cells, they kind of hide between other cells. Is that correct? Where they kind of hide or kind of camouflage themselves in between? Yeah,
they kind of camouflage themselves. Yeah, but if you the good news is that if you have the right marker or the protein marker, you can uncover it in a way that it's not hiding anymore, and you can see that distinguished from all the other cancer cells, and you can actually see the spatial location of where it would be hiding with respect to the other cells.